This is a short video on how to use the BLAST function on NCBI database. So suppose that we are looking for similar sequence for HbA2 hemoglobin alpha 2 for humans. If we go to the FASTA sequence for this gene, which is under the genomic category, so let's scroll down to the sequence information for this gene under genomic. This is the FASTA sequence that we want to input the sequence file into BLAST. This is the HbA2 BLAST sequence. And let's go to the NCBI homepage. BLAST, there's a quick link to BLAST on the right menu here. If we click BLAST, we can use the nucleotide BLAST because we're just looking for a nucleotide sequence. If we're looking for protein, then we use the protein sequence. If we're looking for translated nucleotide and search that against protein database, then we use BLASTX. For protein sequence, if you want to input a protein sequence and see which is the translated nucleotide, then we use tBLASTN. If we're looking for translated nucleotide sequence against the translated nucleotide database, then we use tBLASTX. Okay, so let's start with nucleotide BLAST since we have the nucleotide sequence. We can either enter the accession number, which is, if we go back to here, okay, so this is the accession number for the genomic sequence. But since we have the FASTA sequence, so we just copy and paste the sequence onto this query box here. Underneath, we can select which database we want to search this sequence against. So if we select human genomic and transcript sequence, then we are only looking for human sequence. We can click for others, or we're not limiting any kind of genomic sequence. Then we'll be looking at all sorts of species that have sequence that are similar to the human HbA2. Okay, so suppose we're looking for anything in the mouse genome that looks similar to this human sequence here. And we can optimize the search results by selecting for highly similar sequence. Other options are for less similar or somewhat similar sequence. Okay, let's go for BLAST. And this usually takes a few seconds to a few minutes because we're looking for the mouse genome, not the human sequence, so it takes time. So now it says there's nothing that's similar in the mouse. So that makes sense because we're only looking for, because our input is the human sequence. Okay, so let's go back to BLAST again, and let's say we are just interested in human sequence. Okay. For mouse sequence, we can still get something similar if we select somewhat similar sequence here. Okay. So let's go for humans and see what the search results would be like. This also takes a few seconds. Okay, so this is the results, meaning that we've got matches. Okay, if you mouse over these lines, it tells you all the results that are similar in alignment to HbA2. Let's look down here. The lower the e-value, okay, that means the identity is 100%. Okay, we can see that some sequence that are similar to HbA2 underneath the top two results. So these sequences also contain part of the sequence that are similar to HBA2, but they're not exactly the same. The first match would be what we are looking for. Okay, we're confirming that the sequence that we entered is HBA2. Okay, so this is how you can use the BLAST to look for exact matches or similar or somewhat similar sequence that are almost identical by using sequence alignment in the BLAST function of the NCBI database.